Intro music. From Microbe TV, this is TWiEVO. This Week in Evolution, episode 105, live streamed on August 21st, 2024. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Great to be together. TWiEVO 105. And man, look at these upgrades. We're in these.
Fancy, huh? Got a
Yeah,
home
I've got
frame.
my.
We got your
Woo,
name and your affiliation there.
mhm. I'm liking it. I feel like, the level of professionalism, we're going to have to up our conversational game too here somehow, Vincent, as we march through
Yeah,
some
we
fun science.
Nels, we can't be too thorny, right?
Oh, yeah, there you go. We're already off the rails here. We're back into our old patterns. You can dress up a podcaster, but I think the conversation is going to be about the same. But hey, great to be together. Great to see you. And 105. I think you already gave away a little bit of our
Yeah. I want to
topic.
thank our moderators for joining us. We have Les here today
Good.
from California, Barb Mack from the UK,
Wonderful.
Andrew from New Zealand,
Hmm.
Steph from San Francisco. Thank you all so much for joining us.
Oh, yeah.
And tell us where you are from, folks. We'll go through that
Yep.
before we get talking about science. But this is Wednesday, the 21st, tonight at 8 p.m.
Hmm.
Office hours, special guest, Dixon Despomier. That should be fun.
Yeah, that's a good one. That goes back to the origin story, right? Of This Week in Virology when you and Dixon started
Dixon and I
that.
started it, yeah.
Yeah, from some years
And
ago, and here
and
you are.
here we are with what 9 or 10 podcasts, and I was given Nels an award at ASV up in Toronto. I think it was 2015, maybe
That sounds right. Yep. This was in London,
London, Ontario, right?
Ontario, Canada, and the American Society for Virology meeting where we got our start, our origin story for This Week in Evolution. That's right.
So we were at a bar. I think it was called Toboggan,
That's right.
and I said, Nels,
Yep.
you're
Mhm.
the evolution guy. We should do TWiEVO. And you said, let me think about it.
I did. I think I said yes, but thinking that, maybe you called my bluff, maybe is one way of saying it, right? Because then a couple weeks later, here you were
Yeah.
showing up on the emails and saying, let's get underway. And my goodness, here we are, more than 6 years later and more than 100 episodes in. Really
Yep.
enjoying,
Yep.
really enjoying the action and the live streaming, where we're bringing in folks from all over the world, which I think has a really beautiful kind of parallel, Vincent, in the work we talk about, the papers, right, that we lift up, and when we look at the author lists, there are people working at places all over the world, and here are our listeners from all
Mm-hmm.
over the world. It sort of has a nice echo on the conversation, sort of how science is practiced and conducted. In this case, evolutionary ideas and
Yep.
concepts and advancing them with experimental systems. And then our conversations, which are also highly international. And so I can't think of a more fun way to do it.
Speaking of international, Philip, oops, we have to fix that. Philip is in Wales, UK.
Welcome.
Abdul Aziz is from KSA, Kingdom of Saudi Arabia, 38
Good to have you
degrees
back.
Celsius. Here we
Mhm.
have a little
Woo.
bit of a cool spell.
Mm.
In fact, yesterday it cooled off. Today, right now, it's 22°C. Wow.
Wow. Yeah.
And it's one of those August cooling offs. It makes you think fall is here, then all of a sudden it gets hot again.
Exactly. First cracks in the armor of summer in this location as well. Although we're checking in a little north of 30 C, I think today, but then cooling off in the days ahead.
FSM is the dog is from Simru. Must be Wales, right?
Mhm.
You think?
Yeah,
I'm
well, the
guessing.
weather is consistent with that hypothesis.
Mark Wheeler is in Helsinki.
Great, welcome
20C
back.
and sunny.
That sounds beautiful.
Pete is in London. He's given us half an ear.
We'll take it.
Jeremy is in a Seattle-ish overcast kind of day. Very cool.
Nice. Welcome, welcome.
Is if you think my audio is too low, tell me.
Mm,
I can crank it up a bit. Last week it was too high for the live stream, so could be you.
yeah, always
What
adjusting as we go.
what I learned today is in India where it's humid and hot.
Well,
Not
yeah,
surprising.
welcome. Yeah,
Here's today's paper, by the way. There's a link to it, courtesy of Les. So you
yeah.
can check
Thanks,
that out.
Les, for putting it up there. There's a great, at the very least, there's a great kind of summary, both figure, some structured abstracts, etc. I don't know if the paper's open access though, Vincent. Were you able to? Um,
No,
I,
it isn't open
yeah,
access. No.
which is a little bit odd. It should be. But anyway, there's the link. If you want to go, if you're as part of our conversation today or afterwards, if you want to go deeper and you don't have access to that paper, reach out to us on one of our channels, email, DM on Twitter, whatever it is, and we can get you very happy to send along a PDF copy of the paper.
Okay.
Yeah,
All
yeah,
right. So I just cranked up my volume here.
I can
If
hear it.
hello, you can hear it go up, Nels. Yeah.
Yeah.
So
That
folks,
was a
tell me if
marked
that's good.
difference.
I do have some more leeway here, but let me know. Let me know what you want to do. Let's see what we have less. Okay, that's the link for today's paper. Unfortunately,
Yeah,
behind a paywall. Claire is from the UK. Welcome.
welcome.
Tracy is in Chapel Hill, North Carolina. Okay. Barb Mack says the sound is better
Better.
now. Very
Good,
good.
good.
We're at I have it at 100% on the
Ooh,
interface. So there you go, Tracy. Welcome. Uh, Simru is Wales in Welsh, so it's is Wales, not in Wales. Okay. Thank you
there you
very much.
go.
That was close, right?
Yeah, we're in the right ballpark for sure.
Abdul Rahman is in Brooklyn. Chilly.
Awesome.
Yeah, it's 22 in Brooklyn too.
Ooh.
It's a little chilly up here today, and
Mm.
would like. Oh, here's how to pronounce Cymru. So totally, Cymru is Cymru.
Yeah,
Yeah, Wales has got that tough language, Welsh, right?
correct. Yeah, I've run into some snags with some authored last names from that geography in the world. Not
Rami
intuitive.
from Kyrgyzstan. Well, that's far away.
Welcome,
Welcome.
welcome. Fantastic.
Oops, I just turned
Ooh,
myself off. You
yeah,
think I
come
can
back, Vincent.
figure out how to
We
get
need you.
myself back?
Feeling kind of lonely over here. Uh, a lonely guy podcasting. That'll come up in our paper today. Another
Okay, let's
preview
see if
there.
I can. All right, so I got. Oh man, I screwed it all up. All right, I gotta bring this back. Now the whole thing
Whew.
is a little bit messy. So now my fancy thing. Where is my camera?
Yeah, you're on the
Stream.
background.
It's supposed to be. There we go.
There
Is
we
it better?
go.
But where's the background?
Better.
Yeah, I'm on the background. We don't
Hmm.
want to be on the background. So how does that work? So this is me alone with
Oh, looking good. Yeah, look at
background,
that.
and then me and Nels is supposed to have a blue background, but
Mhm.
this camera screwed it up. So how do I get the background? Show in the background. Yeah, the background. Maybe it's
There's the blue screen.
in live messing around. You know.
No, I love it. So, Vince, there we go. We got it. Woo. I was about to charge forward to try to carve some space, but you solved it.
All right,
Um,
now let me move. Oops, now I screwed it up again. What the hell is with this background shit? Sorry about that. Background has to be below the cameras. There's a hierarchy here. Now I
Ah,
just want to move this whole thing
yeah.
up. And last time I did that, it moved only the background. So how do I move the whole thing up?
It looked like it
Because
just
that way,
worked. I think you just did it, at least from here.
no,
It looked like it.
it was, I want to put it on my teleprompter because that's what
Oh, I see. Gotcha. Gotcha. Yeah.
but when I grab this blue part, it just grabs the background and takes it off. So how the hell do I move the thing? I know I did it before. Maybe I can like grab the edge, Nels. Interfaces are tough. Now it gets larger. How
This
do
is
I grab?
technology at its best.
Yeah,
I'm gonna.
glue there.
I'll tell you what, Vincent. You keep ruminating on that, and I'll introduce the paper
All
and
right,
the author.
let's welcome a few more people.
Oh,
We
yeah,
have
sure. Yeah, yeah, yeah.
Vanity
Great.
Nutrition is here
Welcome, another
from
moderator. Great.
New York. Jan is from La Jolla, California. um, okay, that'll do it. You can start working on the paper,
Fantastic.
Nels.
Welcome all. Um, Good to have you along for the ride, and keep commenting in that chat, thread early and often, and we'll get to some questions and some conversation as we go. So Vincent kind of gave us a little clue to the paper here. So this is titled "The Convergent Evolution of Plant Prickles by Repeated Gene Co-option Over Deep Time." Paper showed up just a few weeks ago, made a nice splash out there, from Science
Oh,
Magazine, a research article, technically in the plant genetics section, but with some obvious, evolutionary ties. And both Vincent and I are big plant fans for obvious reasons. I think we all are. And so when there's a really interesting, and I would say charismatic, morphology here, the plant prickle, and we'll talk about that, what that is, in more technical or scientific detail. And thinking about how this evolved repeatedly, both was gained and lost. Pretty interesting story, that just came online here. So who did the work? So a big list of authors, as I was hinting at, international team. First author, James Satterley, and senior author, Zachary Lippman. These scientists are at Cold Spring Harbor Laboratories, not too far from your location there, Vincent, but out on Long Island, New York. But really, a team coming from all over the world. So we've got Valencia, Spain. We've got Johns Hopkins in Baltimore, Edinburgh, UK, getting over into that area where we've got some listeners, some participants today. Not only universities, but also agricultural research organizations in places like Israel, Germany, as well, plant crop genetics departments, Martin Luther University, New Mexico State, a little bit in my region, my backyard, the Mountain West here, the Department of Plant and Environmental Sciences, Mount Holyoke College in the mix here, museums, the Natural History Museum of London, Toronto, Ontario, Canada, as well. And so really, worldwide effort, I would say, as scientists have come together to sort of contribute. And it's going to show, Vincent. So, in addition to some of the evolutionary ideas, some of the sort of classical genetics approaches, but really the range of this paper, I think, is what kind of speaks to its impact, sort of the ambitiousness of the approaches to move science forward here as it relates to the evolution of plant prickles. And
so this is international, like our audience, right?
exactly. And so, that's kind of the idea here of what what we're doing here is to sort of facilitate these conversations. Not only are the scientists doing this all around the world, but we're talking about it all around the world as well, and sort of breathing a different sort of style of life into the work. But
So we
why
have a
should we?
question that you
Oh,
could
great.
answer
Yeah.
first. What I wonder what the difference between prickles and thorns are.
So there we go. What is a plant prickle? Why should we care about them? Fantastic, Andrew. We're all on the same page here. So first of all, just some definitions. So these plant prickles, I think, are technically defined as sharp epidermal projections. So epidermis, plant skin here, I guess. And this was really a morphological innovation. So it's some intuitive sort of ideas about why plant prickles might arise or be selected on natural selection might favor the presence of these things. So some obvious ones are defense from predators, right? If you've got a prickly morphology across, now we're talking about not just the sort of stems or kind of, those parts of the plants, but also the leaves. The plant prickles are often found there, and they've shown up independently. So this is this idea of convergent evolution that we'll be talking about. So an obvious one, defense from predators, but other sort of ideas or hypotheses with, in some cases, some support have been put forward. So prickles might help with the climbing style of plant growth. And I'm not sure the sort of biophysics of that, but the morphology or the structures are adding some kind of rigidity or allowing growth, promoting growth. Competition between plants. Maybe this is sort of like the sharp elbow hypothesis. If you're a prickly plant, it might be harder for someone to grow right up against you if these prickles are actually causing physical damage to potential competitors, other plants that might be sort of vying for those limited resources or stretching towards the sun to gain the ability to do photosynthesis. And water retention. So prickles probably have some role there as well. So you can imagine all these sort of positive scenarios, but also, are these things, also costing plants? If you invest in prickles, this is a developmental process, you're putting resources into these prickles, and so it could also be the other side of the coin is prickle loss, and this will come into play. It looks like we have a botanist in the audience, and getting to Andrew's question, right? So here we go.
So FSM is the dog says in the common language, the terms are more or less interchangeable. But in botanical terms, thorns are derived from shoots so that they may or may not be branched. They may or may not have leaves, and they may or may not arise from a bud. Spines are derived from leaves, either the entire leaf or some part of the leaf that has vascular bundles inside, like the petiole or stipule. And prickles are derived from epidermis tissue. There you go.
There you have it. And so we call, in the case of roses, every rose has its thorn, or in this case, not every rose has its thorn as we go into the work. Those are prickles. But in other cases, so citrus and honey locust, those are true thorns, as we're pointing out here, grown from these branches. And I guess spines, this would be like cactus spines, right? These are modified leaves, is my understanding here. So, and the prickles are located near another sort of charismatic structure among plants, the trichomes, these hair-like structures. And so somehow that might be patterning or, the morphology here. I think a lot of open questions actually about sort of how this development works. And the authors here will bring in a new gene or encoding a protein, a molecular player in this process. Okay, so that's sort of the plant side of the menu. I would say the evolutionary side of the menu is this concept of convergent evolution, which we've talked about a bunch and is fascinating and continues, I think, to be a really useful concept for moving kind of evolutionary biology, experimental biology forward, in this case, through a genetic lens. And so that's kind of one of the core questions here, is that when you look at all of the sort of appearances of prickles in different species of plants, whether it's an eggplant or a rose, is the genetic program the same? Are the same genes encoding proteins involved in the presence of these prickles or the development of them? And so, the authors sort of use what's already known, I think, to make a really clever, choice in where to study this to get a better genetic foothold on the basis of prickles in the first place. And so part of this, as we talk about a lot, Vincent, involves sort of human influence on the evolutionary process, human selection. So you can imagine in a lot of plants like eggplants, tomatoes, and potatoes, the Solanum genus, that humans have probably selected for on many occasions the loss of prickles, as we're predators here trying to get around sort of the negative impacts of the physical damage a prickle can do versus the edible fruits and related parts of the plants that we consume regularly. as a species in the domestication of all a lot of these different plant species. And so a kind of a well-known example that I learned about from the authors is the brinjal eggplant, a variety of eggplants that was known sort of historically to have prickles, but have lost them. And then other botanists or plant geneticists had mapped that phenotype, the loss of the prickles, to a single genetic locus. And so now we're going to kind of get into some standard classical genetics here. So, and that's where the authors start. So, what was known about this eggplant variety was that there's probably a gene mutation on chromosome 6 of the plant genome. It's a so-called Mendelian trait. So, if you sort of zone in on the genetic location, the loss of the prickles is sort of a result of a mutation. If it's a diploid genome, there are 2 copies. If you lose both copies, you would lose the prickles just because of that single sort of genetic insult, so to speak. And so that's where the authors start. Actually, they do some more mapping, and so by that, as geneticists, what we mean is that we have basically the tools to do breeding, to allow recombination to happen as you do selective breeding, and then you look for markers that will sort of be co-inherited and give you, the markers will give you a clear idea of what genomic regions you're working with. Usually in sort of the modern era, these markers are genetic locations that you can just sort of easily genotype. So you can say, okay, well, this, if we do this experiment, this sequencing of the genome, this marker is coming along. It has this, set of nucleotides or DNA bases. And then what you're trying to do is associate that with the trait that you can see, in this case, the loss of prickles. So they breed a lot of eggplants of this variety, both that have prickles and don't, and then try to map at more fine scale resolution, where is that gene mutation that means that predicts that there won't be prickles there.
So that's a great experimental tool that they have with and without prickles of the same plant, right?
Yeah. And, that takes a little elbow grease, by the way, to do. I didn't go deeper into the methods, but that takes real effort to map something down. In this case, they were able to narrow it to 100 kb region, so 100,000 DNA letters, which is, I don't know off the top of my head the size of the eggplant genome, but that's a huge reduction in the complexity of the problem. And then as you sort of analyze that roughly 100,000 letters,
1.2 gigabases.
there you go. So yeah, imagine how
Big.
you've simplified your problem. If you want to know what is the gene change that's responsible for the loss of prickles, to go through 1.2 gigabases into the billions of bases down to just 100,000 letters. And then it gets even better. So you can, by annotating that part of the genome, what they can say is it looks like there's 10 genes that are encoded within those 100,000 letters. And then from there, to try to use information that they already have to make a prediction about what is the gene. So that's 10 candidate genes where a mutation might be responsible for the loss of prickles. And they immediately, from looking at that list, I think there was a false start. There was another gene that's just outside of that region that another group sort of proposed, but they don't see evidence of that segregating with the phenotype, the loss of prickles. And instead, they point to one of these genes called Lonely Guy, which encodes we'll get to this in a second, which encodes a protein called cytokinin, or part of that biosynthetic pathway. Cytokinin is a hormone involved in plant cell proliferation and differentiation. And so that's kind of, they're using a combination of what's already known about that pathway to then sort of promote that or prioritize that candidate gene out of the list, because it's kind of a intuitive or hypothesis or idea that this hormone could influence, if you express these genes or put this pathway into play, it could connect to the developmental biology of forming a prickle at that location near the trichomes on the epidermis of the plant, the surface of the
They're
plant.
kind of lucky that they saw changes in this gene, because there was another gene nearby, right, that had been previously proposed to be involved, and there were no changes in it.
Yeah. And so you can imagine as a geneticist, if we kind of rewind the tape, sort of going down that dead end pathway, they probably did some experiments to try to see if, there
Mm.
were differences in expression, et cetera. And then as you kind of march through the candidate genes, depending on how you're prioritizing that information, it can still be sort of a puzzle that you're solving as a geneticist. So before we get into why they think this gene lonely guy is so important in the story of the evolution of plant prickles, why was it called Lonely Guy in the first place? So I did a little bit of detective work, meaning 5 minutes of Google searches. And this was first identified, this hormone, in a mutant rice strain. So there's tons of rice genetics that's done for obvious reasons. This is a fundamental domesticated plant, a crop in the sort of rise of our species and key player in feeding the world. And so, and I don't know the backstory, but there was a mutant rice strain that was named Lonely Guy before they even knew
Mm.
what the genetic basis of it was. That it was a mutation in this gene, a cytokinin encoding gene. And so, what geneticists commonly do, you identify or you create mutants. You might treat them with chemicals that promote mutations in the DNA, and then you look for interesting phenotypes. And then that's kind of where Lonely Guy came from. So in this case, rice, as it develops, its spikelets, there are all these, the little what will become the rice grains are, and often in these domesticated species, are in sort of packets of several spikelets that come together. Lonely
Mm.
Guy, they saw just one, and so that was that lonely sort of developing rice kernel that gave the mutant its name. And then that kind of carries along. They were able to identify in the rice mutations a mutation in this family of genes, also shortened to the alphabet soup of LOG, short for Lonely Guy or log, and we'll probably use that going forward to to talk about log in the story of plant prickles.
By the way, Nels, we you already talked about this, but I want to show the picture of these prickles in different
Yeah, yeah,
species, right? So
good idea.
this picture, because these prickles have emerged over many years, as you said, 400 million years of plant evolution, and
Mhm.
here is the one Solanum, right,
Yep. Yep.
species, and you can see they're very long and thin, right? And look at all
Yeah.
the other ones. Maybe the rose is what most people are familiar with, right? You can see the typical rose thorns, but then other plants have. Look at this Saibite has big
Isn't that
thorns,
cool?
and then
Yep.
the Bixa and the Datura have
Mhm.
thorns on. What looks like they're fruiting bodies, I guess. Astrocarium is very different, also on the stem, full of
Mhm.
thorn. You can imagine an animal will never touch that, right?
That's right. Yeah,
So
and so
a thorn is not always the same,
not
and these are all examples of convergent evolution. They're all
ex-
thorns from different places, right?
exactly. And then so the Solanum species, and I'm not sure what species or genus, I'm not sure what species they're showing of Solanum in that picture in the upper right corner, but that's the one that they really focus on. It includes eggplants, which is where they really move the genetics forward here in this prickled versus non-prickled eggplant varieties, the crop cultivars. But you're saying, as you sort of look at this diversity, and this is why they really promote this phenotype, the prickle, as an interesting example of convergent evolution. So all of those different forms below, there's different morphologies. There's needle-like prickles
Mhm.
versus the broad-based ones. That's kind of, I think, a fundamental difference between sort of the broad-based rose thorns that we've all been probably have done a little bit of bleeding at some point or another because of maybe around Valentine's Day versus the needle ones, which in some cases can be invisible, right? It's like you pick up a plant and
Yep,
you notice a minute later your palm is tingling because there's these needle-like prickles that
yep,
have
yep,
gotten to you. Yeah, and so that's kind of one of the I think really fun things about this paper is sort of both sort of glimpsing at all of that diversity, and then as we step through the work, we'll see how they bring in some of those other species to make some of their evolutionary sort of predictions about how the convergent evolution played out here. Independent evolution of the same sort of outcomes, and asking if that
yep.
extends from all the way from that morphology, what you can see in those nice picture snapshots down to the genetic level. And I have to say, Vincent, that idea of thinking about convergence at these different scales, not only what we can see, but at either the cellular level or the genetic level, that's kind of what got me into this game and sort of drives a lot of the work that we do, is using these sort of convergent sightings and then asking, how did evolution solve this or sort of create this the same way or
Yep. Yep.
independently? And if so, how and at what levels? I think that, they've found a really great system to sort of probe some of those ideas. Okay, so Lonely Guy, I should say, I didn't mention, but it had nothing to do with prickles. So it had to do with meristem maintenance, which then led to the sort of morphology of these spikelets, whether you would have the development of many um, grains or grains of rice or just a few in the mutant. But so this is part of a family, the log family of these cytokinin hormones. And evolution's been tinkering with that. So there's a lot of species where there's been duplications of this gene, and this will come into the story in a minute. But before we get there, so they've identified, a mutation in these eggplant species in that log gene, the lonely guy gene, in the prickleless versus the closely related prickled eggplants. And that's sort of, the first foothold to really go after it. But then what did they see? How did they sort of convince themselves that they are on the right path here? And so luckily there's been tons of eggplant cultivation that's gone on. So they quickly were able to look at the genetics of 23 different ascensions or 23 different varieties of eggplants. Where there are cases where they've lost their prickles, and they see that in 22 out of the 23, that same mutation. So it's a splice site mutation, meaning that as the gene is being expressed, the DNA is being converted to RNA. The exon intron structure, there's a mistake where you don't take out one of the introns, and then this sort of brings the sort of gene expression off the rails. You're not able to translate those transcripts, the messenger RNA into protein. You now have, because you don't have that protein, that hormone, you're now not able to do this developmental biology. That's sort of the idea here. And so in 22 out of those 23 varieties that lack prickles, they saw that same mutation. And so that's important because it's sort of what it implies is that mutation happened once, and then in the 22 varieties that have been selected since, you don't have prickles. Now, in the 23rd, there is actually a different mutation. There's a 474 base pair deletion in exon 6 of that gene, a different way to go to sort of take that gene expression off the rail so that you don't make that protein. And so that's sort of, in a sense, that's the exception that proves the rule, right? There were two ways, even among these it's the same species, even just these closely related varieties, where in that same gene, you see different mutations that get you there. And so I think with that kind of knowledge in hand, they're sort of off to the races. It's looking really good for that diversity of the eggplants. When we think about that recent evolution, even, two kind of convergent losses here. And now they can, start to look more broadly across some of that diversity of life that you brought up from that figure that they show in those little snapshots of different roses, et cetera, and ask, okay, do we also find, if we look at those log genes, do we also find similar mutations? It would be independently arrived at in different mutations, but it might be in the same gene or pathway. And so that's where they start to explore even further. So now that they're a little more confident, like they're willing to invest some resources or try to convince science buddies from around the world, colleagues, that this is worth chasing because they're onto something. They've really linked this genotype or the phenotype, the loss of the prickles, to the genotype, this mutations in this one gene. So they actually go ahead and they sequence and assemble new genomes of different eggplant species, and identify different mutations, loss of function mutations in log genes, in that case, predicted functions that start to make you think this didn't just happen in these highly related ones, but even in different species now of eggplants. About half of the 30 species of eggplants they looked at, 14 out of 30, independently had similar mutations, single point mutations. And so this again gives you kind of those breadcrumbs or clues. Maybe it was pretty easy for evolution to kind of tinker with this phenotype, the prickles, they could be easily gained, or so far the evidence is really pointing to they can be really easily lost. And so, I think as we get into a little bit of podcast peer review here, the title of the paper is Co-option of these log genes for prickle, the gain of function, how to get prickles in the first place. And the notion that this has happened, multiple times as you look over the millions of years of evolutionary divergence. But so far, everything we've talked about has been a loss, which is a lot easier to do. It's a lot easier to eliminate something than it is to create something, biologically speaking. And so, not quite bait and switch, and we'll get into the co-option of the genes in a minute, but that's a pretty different process here. And as an evolutionary biologist, it's really easy to lose things. And so some of that kind of impact from the discovery is like, from an evolutionary standpoint, it's a little bit like, hold on a second, is this really kind of, are we really delivering on sort of what was promised from the title in the abstract of the paper? But, stay tuned, because there's more to the story. And the advance to actually take that, that gene or that hormone and connect it to this biology, I think really is impactful and sort of the meat of the paper here. So then, Vincent, as a geneticist, what you really want to do, in addition to, in this case, sort of citing this natural variation as an evolutionary geneticist, as sort of a laboratory geneticist, you want to see if you can do the same thing. Can I come in with an intervention, a specific genetic tweak that will recapitulate the phenotype, in this case, the loss of prickles? So they, of course, borrow from immunity, host pathogen interaction. All good genetics somehow runs through viruses, as you and I know. And so they're using CRISPR, a bacterial defense system that's become famous over the last decade or so, because it's been repurposed as a tool to make to edit the genomes, not just of bacteria, but of all kinds of species, to then ask whether, the mutations that you might spot in the wild actually have an impact in the laboratory. So they use CRISPR to disrupt the log gene using these sort of standard techniques in eggplants, and see that if they experimentally make that same change under the thousands of changes that you might see in these candidate regions, but across that whole gigabase plus genome, they see that if they just use CRISPR to disrupt this one gene that's kind of recapitulating that same mutation, they lose prickles. And so again, a really great sort of evidence supporting the central role of cytokinin hormones in the production of these prickles. So far, pretty standard genetics, the ability to take a phenotype, identify the gene responsible for it. And these are geneticists. So I think they're a little bit evolutionary curious, but they keep, just as you want to talk about, well, where did these prickles come from in the first place? They go out and they just find more losses. So, now that they've got this in hand, they start to look at the literature and see whether there's sort of, again, clues that this pathway is so important for the prickles. And so there's mutant alleles of log that have been identified in rice and barley. So the original lonely guy one doesn't work on prickles at all, but other log genes. So turns out the sort of gene family. So far we've been talking about orthologs, the same gene in a different species, and that diverges from a common ancestor. But then this family of genes, sometimes called by geneticists, evolutionists, paralogs, is the growth of the family of the genes within a species. And so there's multiple log genes, some like in rice that are important for that meristem development, but it appears in both rice and barley, also ones that can, if you have mutations, mutant alleles, that lead to the loss of barbs, which is the equivalent of a prickle for those crop species. They keep going. So they extend this to Chinese dates, giant spider flowers, which is a cousin of Arabidopsis, the sort of famous geneticist's model plant, and also roses. So not every rose has its thorn. There are thornless cultivars where, again, humans have sort of intervened. There's obvious reasons why, you'd want to have roses that don't sort of like cause you to risk losing blood every time that you're trying to sort of cut off or make bouquets of them. And there they find, again, independent losses. So big gene deletions in some of these rose species, these thornless cultivars, where there's big regions that are deleted, not just point mutations, but the log gene that they're proposing or lifting up as being important for prickles, it's not even there. And so that kind of is all consistent with this story, the central importance of this, not just in the eggplants, but sort of across the plant tree of life, so to speak. They also see other cases where actually it looks like the log gene is there, but the expression of the protein for some reason is down. They don't have a sort of exact way of testing that or sort of connecting what the genetic insult is, but the outcome, less protein, and correlating with the lack of prickles is there as well. And then Vincent, they turn to another virus, like all good geneticists do. This is the tobacco rattle virus.
It's great.
Yeah. The virology among plants is absolutely spectacular. And so this virus has been domesticated as another tool to change the genetics of some of the plant species, including roses. And so they're able to use this system to silence a gene. So what you do is you put in sort of a copy of that LOG, that log gene. You deploy that in the tobacco rattle virus, which will infect these rose cultivars, which do have spines, do have thorns, do have prickles. And what they saw is that in 2 out of 14 of the strains that they recovered, that they infected with this virus that would silence that gene, that they saw big reductions in the prickles. So it's not 100%, but the fact that you get that sort of specific of an outcome is again sort of all kind of the chorus of genetic evidence is all kind of singing in harmony that this is an important part of the genetics behind prickle biology.
Why do you think it was only 2 out of 14? What's going on?
Yeah.
Was the suppression not sufficient, or is there something else that's complementing it, do you think?
Yeah, great question. And they don't go into the details, but I think you could imagine all of the above, that just sometimes that virus doesn't infect as effectively in the cells to have that sort of, broad effect across the stem of the rose. There's still, and in some of these cases, it's reductions. It's not complete loss. The mechanism here, so you're using the homology between the gene that you're deploying, the log gene that you're deploying in the virus genome, to find the complementing RNA sequence of the gene being expressed in the development of the plant. And if that doesn't match up right, or it matches up at 50% versus 20%, that could, not quite tip the threshold to altering that developmental pathway. The timing's important. So the prickles, right, these prickles aren't sort of showing up late in the game and in adult plants at just any time. It's as the plant is growing. And again, remember the time frames here, like plant geneticists, these are patient people. Yeah. you got to let a lot of time happen for these things to grow, over weeks and months, not just over days. And as geneticists that have, like myself, that have worked on microbial populations or even animal cells, sometimes we get a little bit impatient. We want our experiments to sort of happen overnight almost, or within a week instead of months or years. So yeah, I think the fact that you see anything at all is actually good evidence here. So the genetics, I think, is spectacular. I think they've done a really convincing job of saying that in a variety of these species, now spreading over hundreds of millions of years of divergence time from a common ancestor, that this gene, this log gene, in particular, this sort of duplicate, is probably involved in the prickles, because when you take it away, these things go from being there to, in a lot of cases, either reduced or completely lost altogether. But that doesn't quite scratch the itch of the title of the paper, which is the convergent appearance of these. How did these things get started in the first place? Again, a much harder question to ask. And, maybe to preview that, I don't think we're going to get all of the way there, but I think they have, some compelling evidence that they're on the right path in thinking about the origins of prickles in the first place and the involvement of log genes. And so from an evolutionary standpoint, the idea here would be, that the law, so prickles aren't found in all plants, that appeared later. It was sort of an evolutionary innovation that is involved in protection from predators and other sort of possible adaptive explanations that we hinted at the beginning, earlier in the podcast. But, how these things are made sort of from scratch is a really tough problem. Um,
Nels, the log gene is present throughout plants, right?
exactly. And so it's involved
Mm,
in these sort of ancestral roles in meristem maintenance and so, cell development and differentiation. And so the kind of model here would be
mm,
that through gene duplication and then divergence in function, that the basically prickles came as a result of these random experiments in duplications. And we just talked about this recently. We're back to the Ono's hypothesis, right? That the duplication of genes is sort of an engine of genetic innovation. So in TWiEVO 103, we talked about some experiments, I think in yeast, if I'm remembering right. So moving really quickly. Here, the authors are sort of very much borrowing or leaning into this idea by observing the duplication of the log genes in some of these different species.
mm,
And so you've got duplication events and then specialization. And so the way they get at this is they start looking at some more plants and doing some comparative work. They're now looking at the forest nightshade, um, and um, uh, and a few others to see whether are there what's happened in the evolution of that family of log genes, and they see duplication events kind of that they can infer might have happened around the time that prickles were coming online. Um, so, you know, they kind of almost bring us, I feel like, up to the sort of doorstep of kind of the notion of neofunctionalization, those duplications followed by specialization. This could be the expression. And they even say, if they look at some of the log genes where they're expressed, it's near those trichomes as opposed to in the meristem. And so that sort of is, the location where these cells differentiate,
mm.
become specialized. But they don't really go very much farther than that, other than some tree building, um, and so that's where I'm a big fan of this paper and the topic. But I think they haven't really sort of done that same level. They've done an impressive job of sort of surveying all these things, but the same level of analysis on the gain side, they really I think nailed the loss of prickles.
Yeah. Yeah.
um, and that's important because honestly, like, we can be fooled by patterns of convergent evolution. So the the key duplications of the log genes could have happened earlier, and then if you have subsequent losses, basically as you're sort of inferring where you have prickles or don't, the possibility is that there were more prickles earlier, and it just looks like they've come on later independently, but it's a result of all of the loss events. And so that balance between gain and loss, they do a little bit of work there, but maybe not quite as convincing. They haven't sort of nailed the coffin shut on all of that biology. It also
So
gets
now,
messy. Yeah, go ahead.
so is the implication that as the gene duplicates and one changes, so there are different proteins, log proteins. So they
That's
should
right.
be able to look at the differences and pinpoint which amino acid changes could give rise to a prickle and which don't, right?
Correct. So that's part of it. And then has that happened independently? They're saying yes.
Right, right.
Um, and it could, like you're saying, it could be those amino acid changes that sort of change its function, but it can also be expression, right? So,
Right.
and we talked about
Exactly.
this.
Mhm.
Does that protein show up at the right time and place for the prickles to
Right, right.
form relative to, right? And so that's all part of the game here too. It gets messy, it gets complicated because there's sort
Mm,
of, some of these functions as the family is diversifying, or as there's duplicated copies, might have shared functions in other pathways. So it can become entangled. As a geneticist, you might knock out a log gene in one plant, and maybe it affects the prickles, but you've messed up something else in the development of the plant where you can't even really sort of get at that. And so that's where, I think it gets more complicated. And you know, the authors, just as we sort of start to really dig in, I feel like they make a right turn, and fair enough. That's like, it's a good right turn. It's an interesting one. They go back to the sort of what geneticists do. They want to break this, so they actually move um, into an application now that they know the important involvement of the log gene,
mhm,
which is, can we genetically engineer other plants, important ones, um, to lack prickles, which could have a huge impact on domestication, where instead of what has happened, over the course of like eggplant husbandry, where it might take 20 years to find a great prickleless variety that then you pass on and as heirloom seeds, and then you got that sort of farmers over the generations. What if they go back and use this CRISPR tool? Now they're off-roading on these other, they're predicting the log gene that might independently be involved in the original formation of prickles to see if they can intervene there in simple ways to sort of almost proof of principle that any plant with a prickle that you might want to modify to domesticate in some way, that this might be possible. So they do this in 3 cases. The forest nightshade, which I think is an eggplant, basically. The desert raisin, which is used for foraging, I think for agricultural reasons, for cattle or related animals, domesticated animals, and tomatoes, which is an obvious one. Again, if you think about things like the heirloom industry, maybe there's a great variety out there, but it's kind of prickly, and so taking care of that might make it more palatable, more edible. Sort of across the board here. And so, so here they're testing sort of potential overlapping broad functions of logs, but also trying to come up with, sort of a rule book of ways to use the CRISPR system to gene edit to take away prickles. And it worked for the forest nightshade and the desert raisin, not so much for the tomato, but again, that points to, as you duplicate these things and have overlapping functions, redundancy can be built into the system. But so here you
the raisin. Look at that.
Yeah,
Isn't
great
that
one.
beautiful?
Yeah, that's great. So hopefully our listeners can see this. On the left is the wild type. This is the desert raisin as you harvest it from nature or just bring it into the laboratory. And then their CRISPR gene edited version is really lacking those prickles. And so that goes the sort of raisin, the fruit there. On the left in the wild type, you can almost see these little spikes coming out from there, the prickles. Whereas on the right, basically they're gone. And so, if I were a cow, I think I'd be much more interested in eating those raisins on the right than the ones on the left, and what that means for sort of how edible or palatable this forage crop is. So there you have it, Vincent. I think we took a great tour here through.
But they didn't do roses, though, Nels.
Well, yeah. So I think that's right. There are some thornless varieties, but whether, if and how, as they sort of get to the basis of how the log genes are participating there, whether they're overlapping or redundant functions, can you take this sort of genetic shortcut to new thornless varieties? If you could, that would be, that's a huge industry for obvious reasons. Humans love roses, and having that flexibility to, as the collectors, the cultivators of roses, to be able to hit all of the varieties without having to go through that sort of breeding scheme could really unlock, some sort of economic benefits to the industry and sort of increase the number of people who will mess around with roses.
Well, I guess everyone who's handled a rose gets stuck, right? You can't avoid it.
I certainly can't. I've got a couple rose bushes in the front yard of my house, and anytime it needs pruning or anything like that, even if I'm wearing gloves, somehow
Goes right through the gloves. Yep.
Yep. And there are some real, microbial consequences to this. The fungal diseases or sort of infections that can result from this are not trivial. Mm-hmm. And so, rose thorn disease shows up in the clinic a lot. The dermatologists are up against that, and so imagine, those are sort of some of the kind of indirect effects of being able to intervene here into the future of thornless roses. And they've sort of the authors put forward a rule book here for getting there. Now it's like turning back to evolution. This is our podcast is This Week in Evolution. It's not This Week in Rose Thorns. And so that's where, I maybe would put a final note where I've already I don't want to sort of belabor the point, but really convincing that the log gene is important, that you can lose this in simple genetic pathways. Convergent loss here. There are a lot of ways to simply, whether it's a point mutation that messes up the protein translation, all the way to deletion or, related sort of genetic insults, really easy to lose prickles. And you can imagine, I'm kind of curious, in addition to sort of the obvious sort of, biosynthetic investment of them, how is natural selection acting here? And on the loss side, a lot of other systems and work there. On the gain side, less so. And I think that's where the paper, points to some, pretty logical or intuitive sort of the log gene duplicates, it expresses differently. There's some neofunctionalization, but how that fits together and how that tells the story, that's work ahead. And it seems like a great system. There's another great geneticist, Vincent, that comes to mind from the location where this work was done, the central location, Cold Spring Harbor Laboratory. I'm thinking about Barbara McClintock and how
Mm-hmm.
she used really dramatic phenotypes, in this case, the kernels of corn and the color patterns, the
Right.
sectoring of it as a geneticist, to link that to these genetic hopping elements, the transposable elements, and to really lift that forward here with the prickles, another sort of charismatic morphological feature, where the genetics are now starting to come into focus, allowing sort of future work to tackle that idea, that really interesting one, the convergent gain of this. How many ways are there to do this? And so here, I think it's maybe I'd close my remarks on this paper by thinking about, again, sort of the sort of continuum of convergent evolution. So is it always the log gene across this whole sort of spectrum? It feels like for some of this morphology, the broad spines all the way, or prickles from roses all the way to the needle-like ones and tomatoes and other members of the Solanum genome, that's true. But are there really independent ways to do this with a different gene family, or is there a different way to that? And so certainly the thorns, the like technically correct thorns that come out of the axial branches as opposed to the plant epidermis, or the spines from a cactus, which are modified leaves, those are very likely not to be the log genes, because you're plugging into different morphology or different parts of the plant to modify a leaf versus to put a prickle up from near the trichomes. And so, that is also convergent evolution. It's just sort of how you frame it. Like the function, I think, when we think about cactus spine in predator defense, et cetera, is probably, functionally the same, but genetically or developmentally very different. And so where do you, as a evolutionary biologist, as a geneticist, where do you draw that line to sort of make these useful comparisons? And so, the authors don't spend a lot of time there. I think they had more than enough in advancing the log gene or connecting that to the prickles and all of these diverse species, but with potentially more to come as we think about these sort of very conspicuous specializations of plants to protect themselves and to sort of influence their growth.
We have some comments, questions here, Nels. Let's
Yeah, let's do it. Let's
go through that. So, Les wants to know, why don't florists create thornless roses?
Yeah, I think they have, with limited success, right? Because so there, as we think about human selection, so we've kind of probably skirted or haven't really talked about this explicitly. Is all of the processes we're talking about, Mendelian inheritance, all of the natural selection here, fits under the Darwinian sort of notion of random mutation with natural selection acting. Then as humans, we've in many cases, as we've talked about on TWiEVO over the years, we've put our thumbs on the scale of evolution as florists, or as we've domesticated both animals and plants in the whole agricultural revolution. And so using before work like this, where we knew that this was the gene that was at the sort of explaining that it's very possible to get thornless roses, but it's not easy. And the trade-offs that you get, right? You might get a thornless variety, but the rose isn't that sort of,
Mm.
eye-catching, or maybe it's not very aromatic. And that's because you're doing genetics in sort of the old traditional way of, well, I really want this thornless thing, but what are the hitchhikers that come along?
Mm.
And so that's where this advance, I think, really kind of sparkles, is when we think about, okay, now that we know it's probably this log gene, go in and mess with that. All of that morphology, the sort of beautiful, colorful rose emoji that we're seeing here, and in real life, the ability, all of that aroma that we have selected on, could come along for the ride, because it's sort of a genetic scalpel in order to get rid of the thorns in those cases. So stay tuned. I'm sure there's many interested parties who are, including probably some of the authors here, who are advancing this even as we speak.
FSM says the charm of the roses is the pain.
Yeah, that's right. And what do as we get our genetic approaches get more powerful, um, I think that leads to some really interesting philosophical conversations. Uh, are there thornless roses that might be completely beautiful and less likely to give us that pain, but it's sort of at some point we lose the sort of the history or the metaphors or the good and the bad coming together in a sense, which sort of approximates real life and is part of the artistry here, I would say as well. And so,
Hmm.
yeah, I like this point a lot.
Pete says, "Have you
Yeah.
watched a goat eat
Hmm.
dried up thistles? Some animals can eat these sharp things, right?"
Well, there you go. And so there's evolution on all sides here, right? So how do you,
Yeah.
if you don't have a CRISPR tool and you're hungry, how does natural selection act over time as, in this case, the goats are eating the dried up thistles. There's some real advantages there. If there's a mutation in the population, that means you don't really care about some of these prickles, but everyone else around you does, whether it's a close relative or a distant one. That ability where you can see how natural selection could act on it quite quickly because you've now just cornered the market on these resources that are
Mm.
completely unpalatable but bring you energy, which ultimately means from a genetic standpoint brings you more kids and very quickly can sweep through a population. Yeah.
We have someone else joining us from Vienna, Austria. Welcome, someone
Welcome.
else. Okay, now a couple of comments about cacti. So Steph says,
Yeah.
"How do cactus spines
Hmm.
fit in?" And FSM says, "Cacti are shrunken leaves to prevent loss of liquids, hence they evolved in deserts."
There you
And
go.
Steen, who is a plant virologist who I know, it
Awesome.
looks like cacti have petiolar leaflet stipular spines rather than prickles per se.
Yep.
So
Exactly right.
good.
Yeah. And how I don't know enough about trichome biology, so I think even for the spines that makes complete sense, or the desert and water retention. For the prickles, which the authors put forward here as one of the several kind of adaptive possibilities, water retention's there too, and I'm not sure if those, that's sort of an interesting developmental question, kind of along these lines, whether the trichomes or the morphology nearby. If you don't go to a prickle, is that sort of are those cells or the morphology around there? Is it more leaf-like, and sort of vulnerable to water loss because water retention is part of the story here as well. That's another, I think, important side note here, maybe general note as evolutionary biologists, as we're looking at these sort of dramatic or morphological phenotypes. It's so easy to kind of imagine these intuitive, so-called just so stories where it's like, oh yeah, it's a prickle, which means the predator can't get there. But if it's a desert and it's water retention, sort of
Mm,
a lot of times
yep.
these are more complicated, or there's no selection at all. It just happens sort of as you pull the marbles out of the bag of the genetics. There's genetic drift. Things get passed on that may not be selected on. It's just sort of the roll of the dice, or the probabilities of something going forward as populations bottleneck and grow and shrink and grow and go forward.
Tom's joined us, our moderator. Thanks. Good to be here.
Yeah,
Good for
great
you
to
to
have you
see
back.
you.
Yep.
So G says, "Hello, everyone. Just watched a Michigander try to grow tomato seeds from
Wow.
1977. They germinated."
Fantastic. I
Very
love it.
cool.
Yeah.
Okay, here we go. This is a good one. Les says, "During World War II,
Mm.
a rose prick infected a British man and it was killing him. He was the first person to get penicillin. It was curing him, but then the supply ran out and he died."
There you go. All of these sort of entanglements with history. Um, and as it relates, so we were just hinting at that too, right? Some of these infections that result from the rose thorns in particular, and the microbes that hitch a ride. There's a lot of fungal species out there who are hoping that we don't go thornless on the roses because that sort of closes off a potential route. Obviously, there's plenty of wild varieties and species out there, so I think the microbes are safe among all of the things that we're doing to the world.
So Tom says cytokinins. I was in Skoog's lab in the 70s. They discovered cytokinins.
There you go, and they show up. This is I think the beauty of evolutionary biology to see how nature is tinkering with these genes and pathways in different ways for really cool outcomes. In this case, the plant prickles. Yeah.
So continuing the Michigander
Mm,
farmer talked about
mm,
drift
yep.
with seeds from 1977. Yep.
Yeah. So the number of seeds that those plants made back in the 70s versus the ones that survived to be germinated today is not all of them. And that gets at that which ones just happen to be chosen, what traits do they have, and then as they germinate and show up and start to breed with modern species today, where you're seeing or getting a hint at all of that sort of complex genetics playing out, um, sort of with the randomness of our ability to keep seeds and germinate them at different times, sometimes decades on, or even I think there's some work recently, maybe out of Russia, where there have been seeds that were preserved in tundra that
Yep.
then have been able to germinate thousands of years on, which is really wild, kind of time machines almost, from those seed stocks.
So Tom says my attendance in live stream limited by commitment to restoration project on our
Mm.
creek property, ecology intertwined with evolution. So I'm posting the link.
Oh, very cool.
So check it out.
Yeah, nice. I've been doing a little, not on my property, but doing some creek restoration in and around Salt Lake. One of the things that humans intervened in my neck of the woods is removing beavers, eradicating beavers from the landscape. And so now, as humans, we're going out and making beaver dam analogs in some of the creeks to try to recreate the sort of temporary dams that allow the sediment to build, slow down the water, counter erosion, and sort of try to replenish some of the aquifers. And so, kind of wild to think about some of these human interventions. The beavers are so much better at this than we are, and yet here we are.
Les says no prickles are their raison d'etre.
There we go.
Okay, Peter says, I wonder if cosmic ray or sun radiation activity has much to do with gene mutations
Yeah,
and modifications that end up changing species or explosions of diversity, as in the Cambrian explosion.
absolutely. This is totally kind of a random thought thread tying into there, but I've got a colleague here in the biology department that just discovered that in zebrafish and other fish, the mothers provide a molecular sunscreen to their offspring. Um, and so, if you follow that evolutionary story, the fish that are living near the surface that are sort of exposed to UV radiation are more likely to encode, the mothers are more likely to encode and then provide these molecules, Gadgisol, to their offspring. And so, yeah, the knife's edge of mutation here, right? Cosmic ray, sun ray, any kinds of radiation. One of these drivers of genetic change and evolution. And again, these sort of double-edged swords where mutations can be, as we know, highly deleterious. The sort of name of a mutation, all the way from kind of a longer evolutionary view to being the grist for the mill of evolution. You create this diversity that then natural selection can act on in positive ways.
FSM asks, would the development of a thorn be the equivalent of vestigiality, selection by circumstance? The first thorn was a broken part of the plant and was an
Yeah.
evolutionary advantage.
Yeah, interesting idea. And that's sort of, I think the authors here spend a lot of time on the loss, or, observing the losses, whether it's in nature, the different sort of the convergence of the loss, doing that in the laboratory with the CRISPR or the virus gene silencing strategies. But they don't talk a lot about the evolutionary advantage of the loss. And so I put forward sort of the obvious thing about just investment, but how the sort of as populations are propagating in different environments, and this could be different predators, different locations, et cetera. How that plays out sort of in the longer term for selection, and in changing environments, how something that might be advantageous at one moment might be deleterious at another. That's all getting litigated in complex ways at the kind of ecosystem levels, in ways that oftentimes we don't know because there's so much complexity, and it changes over time. And so that's another part of, I think, the possibility of this system and these important questions to really understand both sides of the coin, the origins and the gain, but also sort of the loss. And then, even the reversion. Are there cases where populations have lost it, or it's gone down in frequency in the population and then it's rebounded back? And you can imagine now that you have the sort of parts list, the ingredients, the log gene and connected it to this morphology, that in future work, you could start to look over time at the sort of frequencies of these gene mutations rising and falling, and trying to correlate that with the environment or changes in predators, et cetera. And so, yeah, this is great questions. And I think, this paper is a great example of great science, where when you advance in sort of the understanding here at the genetic level, it opens up all these doors to new possibilities. Good science is usually kind of sort of like not satisfied. It leaves you more hungry, even though you've learned something new. You want to really learn more, sort of with that foothold. And I think this paper falls nicely into that category. Yeah.
So FSM also says they use nettles for cheese, Northumberland nettle, for example.
That's right. Yeah. And we're seeing, as we're getting a little more adventurous in our salads, right? Nettles, I think, are showing up on the menu.
Yeah.
And a lot of times physically removing those prickles in some cases. But, there's another opportunity if you wanted to, have sort of a bespoke salad, kind of wholesaler. If you could genetically figure out how to get rid of these nettles, there again, we're colliding with a whole nother human impulse, which is suspicion of genetically modifying organisms, that somehow, GMOs and all of the controversy there would come into play. But I don't know, maybe removing prickles is a way forward. That's a pretty great outcome, right? That's doesn't seem so controversial, maybe from a nutritional standpoint. But obviously a lot of, so most of the genetic engineering or GMOs have been advanced in the first stages as, alternates to herbicides, right? Or ways of, sort of intervening there. The Roundup herbicide story is a classic example here, with like mired in controversy, for good reasons. But removing prickles might be a little less controversial. I think we could all agree that a nettle salad that wasn't going to sort of sting your tongue might actually be a good thing.
Steen wonders if the different author groups converged on this story, or if they were co-opted into working on it by the lead authors.
Yeah.
Maybe some mix.
Great question. I don't know the backstory on this one, but you're right. Like, a lot of times it's not sort of, some of the collaborations we see aren't sort of single origin events where the senior authors then go and sort of recruit colleagues. It could be, oh, wow, you're working on this. And someone presents it at a meeting, and then they share that information. It's like, oh, well, why don't we team up? And there can be some real advantages there. That's happened in my lab more than once as well. And so, yeah, good point. There's sometimes complicated backstories on how those author lists sort of show up with the final product, the paper that was published.
Rob says nettles can render a salad invalid.
There you go.
He also says fungi exist in a perpetual state of grace.
I love it. Yeah.
And FSM says radiation provides seeds and seed banks with more mutations. Yep. Evolution selects the good ones.
There you go. Yep.
John says, I wonder if naturally occurring sunscreen would have commercial applications, but maybe what we have is cheap to produce already.
Yeah, Gadgisol. I think that's a good question. I'm forgetting some of the details here. So this is microbial, like the origins of Gadgisol are microbial. Obviously, the sunscreen industry is massive, and both using chemicals or moving towards sunscreens that are more mineral based or metal based, just physically obscure the UV radiation from hitting our skin. There might be some Gadgisol in development. That would be, I think, a pretty low impact from a chemical standpoint thing, and whether you can sort of produce it in mass or whether someone's doing that is an interesting question.
All right, that'll do it for our comments in the chat.
Love it. Great comments, questions, all of the above. Good energy here.
I'll do some picks, Nels, to wrap this up.
Yeah, let's do that. So my pick of the week is sort of on the top, still on topic here. It's a link to, this one is open access. This is a perspective piece PLOS Biology, the journal, and it's titled "Trees as Metaphors to Understand Relationships in Biology." And so I think there were two papers that this perspective piece kind of sets up in this issue of PLOS Biology. But it starts to get at, like, we almost take it for granted, I would say, as evolutionary biologists, as evolutionists, the kind of metaphor of the tree, the phylogenetic tree, the tree of life. But I think, sometimes to the detriment of advancing science, we sort of take it for granted. What do trees mean? How do we use them? And so I think this is a really nice, this caught my eye as a way of sort of stepping back and sort of surveying how we use trees as metaphors, or even like hypothesis generation, and how that continues to advance. We haven't, there's something about the tree that feels sort of permanent, or something has this permanence to it. And yet, and I think that sort of metaphor comes along sometimes as evolutionists. When we publish trees, we think about, oh, this is how we're related to that. But every tree is a hypothesis, and it's sort of based on different information. And so a lot less sort of, robust or stable as maybe our brains sort of let on. And so one of the papers that this piece previews is, I haven't had a chance to go deep yet, but is sort of the next in line, sort of proposing ways to features or characters to build trees of life, or phylogenetic trees, to ask how populations or species are related to each other. And so, before the whole genomic era, a lot of the trees that evolutionists were building were based on morphological traits. Actually, if you go down the parking lot from where I'm sitting here in LD Lab Studios, is the Natural History Museum of Utah, which is just playground of dinosaur bones, of fossils. And on one of the walls is a collection of Triceratops crowns, of the skulls or the heads of these things. And it's arranged in a tree based on who has these horns here, who has these horns there, and what looks the most similar. Of course, we don't have any DNA here. These are Triceratops that were extinct hundreds of millions of years ago. And so there are, when we try to think about the relationship between these species, one way that we've done that, and how evolution have done this, is to say these ones look more the same. They're probably more related. Since then, obviously, we've moved into trees based on genes. That's what we saw in the paper we've been talking about today, from genes and from plant genomes, whether they're in other mutations are gained or lost, sort of as we look at that tree, as those relationships go on. One of the papers here takes a different approach, which is to ask about gene synteny, which is the organization of genes in a genome. So if you take 3 genes, for example, and ask, in this species, the order is 1, 2, 3. Does that hold up for every other species in the comparison, or is it 3, 2, 1, or is there different genes? And to use that as a way of trying to think about the relationships. I love this. I've been daydreaming about this for a while, which is sort of returning to the notion of different features or characters to draw trees as new hypotheses. And so the idea I had, inspired by those Triceratops down the parking lot from me, is whether in the AlphaFold era, are some of the protein predictions, or these structures, could these be the molecular equivalent of a Triceratops skull? And could we start to make arrangements based not necessarily on gene sequence, but by sort of similarities or differences that appear in predicted protein structure? So we're just kind of glimpsing at a few projects that are trying to take that into account and see where it takes us. But anyway, trees as a metaphor for understanding relationships in biology, where it's evolutionary or clonal gene. That's the second paper. It gets into sort of cells as they divide, and the lineages of them, which have also been sort of characterized as trees as well.
I like that.
Yeah. Vincent, how about you? What's your science pick of the week?
So I have a little bit of a political pick, but I think this is really concerning, and people need to be aware of this. This is a column by Paul Offit on his Substack called Beyond the Noise. It's called Project 2025 versus the Public's Health. It's part 1, the CDC. And as you may know, Project 2025 is a 900-page document of the Heritage Foundation, a conservative entity, which is supposedly the blueprint for the next administration if the Republican is elected president.
Right.
And among the blueprints are what they're going to do with the health agencies. So they deal with CDC, FDA, and NIH.
Yep.
So this one deals with CDC. And they say basically CDC failed during the pandemic, which is wrong. Okay. It's a fucking lie. Sorry. Sorry. I'm really sorry.
No, I'm with you. I'm with you.
This is so annoying. And they say the solution is to eliminate CDC as a recommender for all vaccines.
Yeah. Wow.
Never again, quote, never again should CDC officials be allowed to say in their official capacity that school children should be vaccinated. Such decisions should be left to parents and medical providers.
Mm,
Folks, if you think this
mm,
is good for the country, waste your vote because
mm.
this is nonsense. This is
Yeah.
going back to the days of infectious diseases killing tens of thousands of children every year.
Yeah.
It makes zero sense, even in a broader framework of I don't want an intrusive government. Leave
Yeah.
public health alone. We've gotten to the point where we're really good and we didn't fail during the COVID pandemic. We
Yeah.
prevented lots of deaths. Yeah,
Yeah.
there were some vaccine side effects, but that's not a failure, folks. That's
Mhm.
just your excuse. So we just recorded a video version of this,
Good idea.
which is going to be
Yeah.
on our YouTube channel as usual next week. But folks, you should be scared
Great.
to death over this 2025 project nonsense
Oh my
because,
God. Yeah.
I mean, it has huge implications for health and science. They want to dismantle all of it. No experts. They said, we don't want any experts. Leave it up to you.
Hmm.
Good luck with that.
But Vincent, how do you really feel about Project 2025? No, I hear you. I'm with you. And, and this was the video that you mentioned. This will be on, is that Beyond the Noise with
Yeah,
Paul
so it's
Offit?
Beyond the
Yep.
Noise, which we do every 2 weeks or so. We
Yeah.
do a
Yep.
video version of his column. We just did this one.
Great.
And
Fantastic.
there are going to be three: CDC, FDA, because they want to dismantle CDC, FDA, and NIH.
Yeah.
So scientists, they want to dismantle NIH. Okay, they want to dismantle FDA. There's no reason for any of this, and this is a minority of the U.S. population with the Christian conservative leaning. Okay.
Yeah, absolutely. Well, and just to say, on the Project 2025, sort of this blueprint for one of the possible administrations here, we're watching this closely. Also, not just on the coast, but here in the Mountain West as well. And so, a couple of things, a couple of spottings just in the last 24 hours, 48 hours of the convention. In the roll call last night, the Utah delegation pointed out, as they threw their support behind Kamala Harris, that Utah is slated to be a swing state in about 10 years. And sign me up. That's kind of why I'm here, is as we, spread out across the country and try to bring some common sense to a lot of different corners, and have these conversations on some of the front lines. This is an important moment. And so, did you catch, Vincent, the speeches last night, the Obamas, as they
No, I did not.
spoke? Yeah, I thought a really great moment. Some great moments there. Maybe Michelle Obama in particular, about lifting up the Harris campaign, but also contrasting that to the alternate, the Project 2025 directions here, and what a pivotal moment. And so I think both to shed light on this, to contrast it, and then to face this moment with energy and positivity about what's possible. As a former Minnesotan, I'm absolutely delighted to see Tim Walz stepping to the plate here, and some of the joy and energy he's bringing in that sort of classic Minnesota style. He'll deliver not just on, I think, a more reasonable way forward, but also a hot dish recipe that has raised some eyebrows in some church basements over the decades. So, yeah, very important, obviously timely topic, and still scratching my head on some of the politicization of some of the public health stuff here, and how that's really being used cheaply to advance some stuff that really has exactly the impacts, the scary impacts, the dangerous impacts that you're pointing out with this pick of the week.
I don't know why you'd want to mess with public health. It has been so successful. And,
Yeah.
it's one thing if you don't want government intervention in other things, but
Yeah.
if you don't want the post office to be government, you want it to be privatized, whatever.
Mm-hmm.
But leave public health alone. It can't be in the hands of medical providers and parents. Okay? If, as Paul Offit pointed out, the vaccine trials for the mRNA vaccines, 800-page document.
Yeah.
Are the parents going to read it? Are the medical providers going to read it? No. Well, they'll go online and they'll search and get all the misinformation that said this
Yep.
vaccine is not good for you. And they'll say, oh, I
Yeah.
did my research, which is just
Yeah.
sheer, utter nonsense. Okay.
That's right.
People are
Yeah.
fooling themselves. And all of
Absolutely.
you
Absolutely.
conservatives who think, conservative is better, religious is better, less
Mm.
government is better. If you think public health is just a throwaway, you're wrong. You want to go back to polio and measles and
Mm.
tuberculosis. We're headed that way if you
Yeah.
adopt this kind of crap. So
Yeah.
I'm very concerned about this,
Yeah. I hear
not
you.
just for public health, Nels, but also for
Yeah.
science, basic
Of
science.
course. Yeah. Absolutely.
A certain party wants to limit the kind of experiments we do for no good reason.
Yeah.
For no
Yeah.
reason, because there never has been an accident. We are in good control, but they say, no, you can't be in control of yourself. And you just have an agenda that has nothing to do with reality,
Yeah,
right?
yeah, yep, yep. No, I hear you.
So,
Maybe I would even draw a little echo of, the paper today, which is what's really important is sort of the time scales that we consider things or put things into context. And so, for the case for this backlash on the CDC, but not seeing that the bigger time frame, where what has public health meant for lifespans of our species over the last hundreds of years versus did
yeah,
we make
yeah,
a little bobble on we should have been wearing masks for this couple weeks of a thing versus another. I think that's like, for me at least, a lot of this kind of traces a little bit back to that loss of context. And so,
yeah,
you know, how do we, and I think part of our mission is to, with all of the this week empire of podcasts, is to really think about context. We spend hours trying to put things into context and to add that, because that's so important. And I would say, as we're faced with all of the just fire hose of information, misinformation, as we're all curious people trying to kind of navigate the internet or different sources of information. I think part of what we're doing here is to try to equip ourselves with these tools of critical thinking, and also of asking for context when we consider something. And where something might be
yep,
sort of on the surface true in a technical way, but if the impact of that in a bigger contextual sort of setting is a very different thing, that's where the things are really out of whack here, where there are parties like the Heritage Foundation, the Project 2025, just stocked full of the former Trump administration and the incoming administration. They're really taking advantage of that sort of hole at this moment to take things out of context, to for political reasons, and to sort of push things into a different direction. I'm with you, man. I hear you. And the good news is, there's I'm feeling coming out of tune. If you haven't already, check out Michelle Obama's um, speech. I spent some time surfing the Republican convention speeches. So I'm doing the comparisons. I'm getting the context here, and this is a mess. This is not easy, but I'm also feeling some real optimism, some real energy, and hoping that things are going to break in a good direction here. But let's all dedicate ourselves to the next several months of what this looks like, and to try to get behind something a little more positive than eliminating vaccines. This feels like a pretty obvious scenario where we're going the wrong direction if we buy into some of this stuff.
So here's the link for Beyond the Noise. Les put it up there. Thank you, Les.
Yeah,
Um,
fantastic.
Thank you, Tracy, for your contribution to science education. Really
Appreciate it.
appreciate that, helping us to continue these
Awesome.
podcasts.
Thank
And
you, Tracy.
FSM says the U.S. is literally on a precipice,
Yeah,
and
that's
this is the
right.
view. It's true. It's
Yeah,
not. It's not rhetoric. It's true.
yeah.
It has
No,
taken many
and
years to get to this point, and we're this is where
yeah,
we are.
I agree. Barack Obama, I think, nicely spoke to that as well last night. That you've got the world looking back this direction and saying, "What the hell are you guys doing?"
Yep.
And
Yep.
and what is, what are and that part of what's going to happen in the next several months is an answer to that question, and what the U.S. looks like, which is a complicated ecosystem of interactions as we talk about sort of global interactions between countries. But the point that the former president Obama was making there is that when the U.S. is in the conversation, to try to nudge things in a good direction, some good things can happen. And so hopefully that starts to weigh in as well. But bad things can happen too when the U.S. sort of sticks its nose in. And so finding that balance is a level of complexity that's really hard to pull off.
All right, time to wrap it up. We're at an hour and a half. Let me thank the moderators tonight.
Yeah.
Les, Vanity Nutrition, Barb Mack, Andrew, Tom was here as well.
Awesome.
Thank you all for Steph, SF. Did I mention you? Andrew, I mentioned. Yes. Thank you very much for moderating. Thanks all of you for coming and listening tonight, 8 p.m. Eastern, which is about 5.5 hours from now, right?
Yeah.
We
Dixon will
have
be on hand. Yeah.
yeah, Dixon will be on office hours. So
Sweet.
if you're able, see you then. Nels, thank you so much for doing this as usual.
I love it. Thank you. We covered all the way from plant prickles to political prickles today, the 4 Ps,
Totally, totally.
and we'll keep coming back with it. So we'll see you all again next time. Thank you, Vincent. Great to be
You're
together.
welcome. So you can find the show notes for TWiEVO at microbe.tv/TWiEVO. If you like our work, we'd love your support. Microbe.tv/contribute. Questions, comments, either here on the stream or TWiEVO at microbe.tv. You can find Nels at cellvolution.org or on X, L Earlybird, and I'm at microbe.tv. The music you hear is by Trampled by Turtles. Let's get that going here. You can find them at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious. Bye-bye.
All
All
right.
right.
Oh, we're still on.